Hello, this is Mr. Marriott with Noteworthy Science. Today we're going to be doing the standard level learning outcomes for subtopic C2.2 Neural Signaling. Under the curriculum matrix, this falls under theme C, interactions and interdependence, and under the level of organization of 2, cells. Some guiding questions to keep in the back of your mind when you're learning this subtopic is 1. How are electrical signals actually generated and moved within neurons or neural cells? And how can neurons actually interact with other cells inside the body? In standard level, there are 7 learning outcomes that we'll be exploring. Let's begin. So the nervous system comes from the idea of a neuron. So the key building block of the nervous system is a nerve cell or a neuron. A neuron is a specialized cell that makes up nervous tissue, right? And actually, if you look at the this link right here, it's actually phenomenal. Here's actual images of the nervous system, right? And it looks pretty amazing and pretty complicated. And the big things in this topic is all about, 1, how do electrical signals pass along this, but also how do these cells interact with each other, right? Because your nervous system isn't just one neuron, it's how do neurons communicate with each other. That's really all about the nervous system. So let's first talk about just the basic anatomy, if you will, or the different parts of this specialized cell of a neuron. So if we look at a single standard neuron right here, what are the different parts? Well, here should look quite familiar because here is going to be where you're going to find most of the organelles. This is where your nucleus is going to be, the rough endoplasmic reticulum, mitochondria, blah, 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 whatever. This part right here is called the cell body or the soma. And then branching out from this are all of these things called dendrites. So this is what's going to receive a signal. This is what's going to receive the impulse. And then it's going to propagate in this direction along this long skinny part called an axon. And then the axon is going to terminate into these other little branches called axon terminals, right? And a way of thinking about this is the train terminal or an airport terminal is the end point of a path, right? So signals are always going to propagate in this way, from dendrite through the soma, down the axon, into the axon terminal. This means that if this signal is then going to get propagated from this neuron to another neuron or maybe another cell, they're going to now leave the axon terminal and go this way. And that process that we'll talk about is something called synaptic transmission. You should be aware that this signal is not going to be bidirectional. It's only going to be propagated from dendrite to axon terminal. And you don't have to take my word for it. We'll explain why that is. I'll give you a good explanation later in this class as to why that signal will not travel backwards, why it can only travel in one direction. Understanding really how this entire system works, you have to understand a very key concept called membrane potential. Now, let's do a little bit of a physics review. And what we're going to be talking about is voltage. So voltage is what? Does anyone recall what voltage is? When you think about voltage, another way of expressing voltage is sometimes described as potential difference or PD. Right. And so that's really it's potential energy, really, right? So the idea is the greater the potential difference, the greater the amount of work that can be done. So when we look at a membrane potential or potential difference, that means that by definition, you have to look at 2 different things. So for us, we're going to look at what is the membrane potential when we consider the inside of a neuron and the outside of a neuron, or the intracellular space versus the extracellular space. If I'm going to ever use a voltmeter, I can't attach a voltmeter in one spot. How am I going to calculate a difference? Right? You have to take it at 2 different spots because that's going to allow you to calculate a potential difference. So if I want to calculate the potential difference across a membrane, I'd have to compare the inside versus the outside part of that membrane. When your neuron is not currently propagating an electrical impulse, it's at rest, then it has a slightly negative membrane potential. And that value usually is listed at around -70 millivolts. That means that the inside of your neuron is slightly more negative than the outside part. How do we maintain this resting potential? The first reason is one of the most important protein pumps in the entire body, and that being the sodium-potassium pump. If I'm doing primary active transport, what does that mean? We are hydrolyzing ATP in order to move a substance against its own concentration gradient, right? Now, this pump is sometimes referred to as an antiporter, meaning that it's moving things in opposite directions. And both of the things it's moving are against their gradient. Understanding what direction these things are moving is very, very important. I'm not going to go into super detail of this mechanism because that is an HL learning outcome. And if you are curious as SL students and you want to know more, then the subtopic B2.1, we go into that in detail in HL. But this is the too long didn't read version here, okay? And that is, I've represented the pump in green. This represents the inside of the neuron. This represents the outside of the neuron. This pump is currently facing, so to speak, the inside of the neuron, and 3 different sodium ions are going to bind to that pump. Then I'm going to hydrolyze ATP. This protein has now undergone a conformational change. It used to be facing the inside of the neuron. It is now facing the outside of the neuron. The ATP wasn't used so that it could bind sodium. It was already binding sodium. It's being used to change its conformation. Now, sodium has now unbound, and look what we've just done to sodium. We moved it against its concentration gradient. We went from an area of low sodium concentration to high sodium concentration. So we've increased the gradient of sodium outside the cell. At the same time, when the protein is now facing the outside of the cell, 2 potassium ions are going to bind. And then, basically, this inorganic phosphate unbinds. SL students don't necessarily need to know that detail. But this is going to resume its original protein conformation. And now 2 potassiums have now entered again against their concentration gradient. So the action of the sodium-potassium pump has moved 3 sodium out, 2 potassium in. Now, how can this contribute to a negative membrane potential? It's an uneven trade, so to speak. You are getting rid of 3 positive things, but you're only taking back 2 positive things. So you are causing an imbalance of charge. Even though I'm moving cation and cation, it's 3-2. So this is contributing slightly to the negative charge. Now, you might be like, is that really enough to get to -70? No. It's going to give you a negative charge, sure, but that's not the only way that neurons maintain this -70 millivolt. Another factor is going to be what are called leak channels. Now again, review, if I use the term channel protein, what kind of transport am I implying? If I'm using a protein channel, then that implies I'm undergoing facilitated diffusion. And recall that diffusion is a passive process, meaning that I don't require ATP, and substances, in this case ions, are going to move down their respective gradients. The hydrophobic core would not allow most hydrophilic and charged particles to pass through. So these are kind of acting kind of like a pore or a tunnel, allowing something to pass through. Now, this doesn't really make any sense. If I have a channel that's allowing potassium and sodium, wouldn't the gradient just be lost, right? Doesn't this just undo all the work that I just did with the sodium-potassium pump? But it doesn't. And the reason is that you have way more potassium channels than you have sodium channels. And so what's happening is that the neuron is a lot more permeable to potassium than it is sodium, meaning that potassium is going to be exiting out much more than sodium is going to be coming in. So We created this original imbalance with sodium and potassium pump. We've now created a high concentration gradient of potassium inside the neuron. These channels all over the place are going to allow a lot of it to just exit right back. Now again, we do have sodium channels as well, but far fewer of them. So this is again leading to an imbalance. We're losing even more positive charges outside of the neuron. Recall that all channel proteins are molecule specific. So an aquaporin will only allow water through. A potassium channel will only allow potassium through. A sodium channel will only allow sodium through. They are specific. So even though it's not like sodium is like, oh, I'm just gonna like join into this thing. It can't. It will not be able to pass through. And that's because of the properties of potentially the amino acids or the R groups within that pore won't allow, either due to size or whatever, for these things to pass through. Finally, as these potassium ions are typically associated with negative stuff within the cell, as these things have now left, you now get a collection of these organic anions within the neuron as well, and that's causing an even more negative charge to build up. Originally, I had a bunch of potassium associated with this stuff. But now, as the potassium has left through the leak channels, and recall, at this stage, these would have a neutral charge, right? Because you have a negative and a positive. But now, as the potassium has left, you now get more negative charges happening here. So for these three major reasons, the sodium-potassium pump, the imbalance of potassium and sodium leak channels, and organic anions like proteins, all of these contribute to the neuron at rest having a negative 70 millivolt resting potential. That's the starting point. We get electrical signals passing through neurons because of the flow of sodium and potassium, cations. So it's really the flow of a charge. So what we're going to be describing is how does a nerve impulse propagate, which is a fancy term for move along this neuron. We're going to break this down into 3 steps. And again, this is kind of like a general explanation because in HL, we're looking at this in much more detail. So to understand these diagrams here, just to make sense of my notes, this represents the membrane of an axon. And so this would be the space outside the neuron, or the extracellular space. This would be the space inside of the neuron, or the intracellular space. How do we propagate a signal? So the first stage is going to be depolarization. Depolarization is a flip. It is now going to cause the inside to become more positive than the outside. If I want the inside to now become more positive, what's coming in? Sodium, right? But we already talked about that we don't have a lot of sodium leak channels. So instead, we have very specific sodium channels, and these are voltage-gated channels. So the idea is that a gated channel means that it is not always open. It sometimes is closed, hence a gate. So there's actually a structure on the protein that will actually close the pore or allow the pore to be open. For SL, all you'd have to know is that the membrane potential has changed. It's become slightly more positive. It's still negative, but we hit a point called threshold potential. When that happens, these gates open. The sodium voltage-gated channels open, and we already know what direction sodium is going to go. It's going to move from outside into inside. If now I get an influx of sodium, what's that going to do to my membrane potential? It's going to cause it to become much more positive. I'm taking in much more cations. Before, the outside, the extracellular space, was more positive than the inside. Now with a sodium influx, the outside has now become more negative than the inside. I flipped the polarity, hence the term depolarization. It used to be more negative, more positive. Now it's more negative, more positive in this direction. So now the inside of the neuron is positive. We are then going to reset it back. How do I then reset it back? Well, the term is repolarization. I want to reset. I want to go back to being negative. So again, if you recall that the inside of your neurons have a high concentration of potassium, how do I make the cell suddenly more negative? I'm going to now open voltage-gated potassium channels. These are only going to be open when the inside membrane is now positive. So normally they're closed, because normally at rest, the inside is negative. But now when the inside is positive, these gates open, and notice now that these sodium gates are now going to be closed. So all of the potassium is now going to rush out, and we're now going to become much more negative. In fact, this happens so much that you're actually going to get to a point that you're even more negative than what you should have been at rest. This sodium-potassium pump is effectively going to reset the boundaries. We're going to get back to our minus 70 again, because actually there's such a large outflux of potassium because of the potassium voltage-gated channels that your membrane potential is actually going to be even more negative than it was at rest. So we're just going to kind of reset it back to rest. How exactly does this cause a charge to move down a neuron? Well, let's examine how all of these work together. Here is my axon, and for context, down there would be the dendrites, here would be the axon terminal. So I'm moving in this direction. So this section depolarizes. That causes now the adjacent section to depolarize, because we're now going to cause this to become more positive. That means this part's depolarizing. Why doesn't it work backwards? Well, this part is now repolarizing. It can't. So we're moving down in a wave, where this is now going to cause this section to depolarize. This is currently repolarizing. This is now at rest again. So this thing is moving as a wave. Sometimes it's referred to as a depolarization wave, is we're moving this down, 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 down this way. If my explanation was not sufficient to help you explain this process, I do encourage you to take a look at this process in more detail at HL. And something else that we'll discuss in HL is something called local currents, and this explaining how one section is actually causing the adjacent one to depolarize much more in detail. So we will explore that more in HL, but as an SL student, you should never feel that HL content is unavailable to you. You can always look at it if that helps you understand it better, and I do encourage that. The next question we're going to explore is how can we alter, or what factors alter the velocity? I.e., how do we make the action potential propagate faster if we want it to propagate faster? What factors influence this? And so there's two major factors that we're going to look at, and both of them actually are physics related. Who knew? Because we're talking about electrical impulses. The first thing that we're going to be talking about is what's called the axon cross-sectional diameter. Now that's a fancy way of saying just how big, really, in terms of diameter is the axon. And I think this one is fairly intuitive. If I have a literal single neuron with this diameter versus a neuron with this diameter, why does the bigger diameter travel faster? It has to do with internal resistance. Let's consider two different kinds of neurons here, and I've shown the diameter here. In the axon with the smaller diameter, there's physically less space within the cytoplasm, right? So there's all these organelles all over the place, right? The organelles aren't only found in the soma, they're also obviously found throughout the axon. So there's stuff all over in the axon as well. This means that there's significantly more internal resistance within this smaller cross-sectional area. That means that the action propagation is actually going to be slower. Because remember, if I have to do depolarization, 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 there's a bunch of stuff in the way. There's more resistance to flow in this case, or more resistance to a charge flow. Whereas if you look at a larger cross-sectional area, you have a larger volume of cytoplasm, and thus there's more space between the stuff inside of the cytoplasm, which means that if, again, I'm doing the same depolarize, 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 there's less internal resistance. Thus, you're going to increase the rate. Where do we see this in real life? Well, squid have what are called giant axons in terms of they have a really big diameter. Now, I know when we say really big in biology, it's maybe not what other people are thinking, but really big axon diameter is 1.5 millimeters. That's actually quite big, especially when you consider a lot of axons typically have a diameter of 0.5 millimeters or lower. So that, from a biology point of view, is huge. Why is this adaptive for squid? Well, they have a propulsion mechanism. It's actually how they move. Squid have the ability to actually do really quick bursts, and that quick burst could either be for very fast reaction, usually to escape predation. To be able to transmit a signal faster to their muscles, it allows faster movement. So this is adaptive. So they've evolved to have fairly large diameter in their axons. Another factor that increases the rate is myelination. Chances are when you look at electrical wires in a circuit around you right now, you're not seeing exposed wires. The wires are usually coated with usually rubber. Now that rubber is acting as an electrical insulator. In a similar way, we also insulate our wires or axons, but in this case we use something that we've already explored in its ability to be a thermal insulator, and that is fat. So we're actually going to use cells that are fatty that actually do not allow a signal or electricity or charges to flow through them. Let's first talk about how this actually develops, and then let's talk about why this actually increases the rate of propagation. So first of all, it's actually really cool. These cells are called Schwann cells, named after the biologist that discovered them. So in an unmyelinated axon like this, you get a Schwann cell, and then what basically happens is it goes and wraps around an axon in this coiling fashion, causing something that's called a sheath or a myelin sheath. So a sheath is a holder, something that surrounds something else. In this case, myelin is going to be surrounding or coating these cells. Exploring a simulation like this really gets you to understand not only how these neurons are interacting, but you can clearly see the different sheaths that are surrounding the axon. So this is what I'm referring to. Now notice, if you look at this axon, that the sheath is not entirely covering the axon. Why would there be gaps? Why not completely coat the axon in myelin? So in this scenario, I have a completely coated axon in myelin, and in this scenario, I have gaps between that myelin. This system might make sense if, let's say, we consider electricity as the flow of electrons. No problem here. But remember, that's not really how our charge is flowing in neurons. It's because charges have to come out and back in, and then that's causing this to depolarize adjacent sections, right? But it can't, because we have a very good electrical insulator. So in this system, it's actually quite cool, is that the only sections that are going to depolarize are these gaps. We get depolarization that causes this now to depolarize the section way down here. So it's almost like the signal is jumping from gap to gap to gap. And this actually super accelerates the rate. The reason I want to ask that question is because this is a common source of misunderstanding. I think a lot of people assume that electricity is propagated the same way as it's propagated through copper wires, where it's just a flow through the conducting material. But remember, that's not what's happening here. It's actually depolarization, repolarization. So it may seem counterintuitive that I'm actually blocking that from happening, but I'm only blocking it in key points. And by having these gaps, it means that depolarization is only occurring at the nodes of Ranvier, which means that I don't have to depolarize every single adjacent section. I can only have to depolarize much fewer sections. So in effect, this system is sometimes explained or described as jumping from node to node. In reality, it's not really doing this. It's not jumping this way in reality, but it means because it's only depolarized depolarize, it's much faster. We'll talk about this more in detail in HL, but I'd say the key part of SL is understanding two major factors that alter the speed of transmission. That being one, a greater cross-sectional area of the axon, and two, myelination or myelinating axons. Let's use this next learning outcome to actually explore that. Statistics. Doing experiments and looking at papers, surely you've come across two different values, that being R and R squared. Now, these two things are not the same thing. Let's start with first correlation. As a review, we just looked at this when we did transport. We discussed the correlation between certain risk factors and coronary heart disease. And we said that if two variables are correlated, it means that there's some kind of relationship between them. But remember, it doesn't necessarily mean they're causational. Further work has to be done in order to demonstrate if they are in fact causational. But correlation really is just a fancy way of saying, are these variables associated with each other? Yes or no, right? So you can have a positive correlation or a negative correlation, right? A positive correlation is as one variable is increasing, the other variable is also increasing. If the correlation is negative, it means that as one variable is increasing, the other variable is decreasing. So there are two different ways that we can quantify a correlation, that being the correlation coefficient, R, or the coefficient of determination, R squared. Now, these two values are similar, but they do not do the same thing. And I want to try to explain to you how they can be used and what the difference between them are. Now, the correlation coefficient, or R, is again something that we looked at when we did transport, where we described that this value can either be positive or negative, and it can range from -1 to +1. -1 being a perfect negative correlation, +1 being a perfect positive correlation. And the closer you are to either of those values, the stronger the correlation is. A correlation, an R value of 0 means there is no correlation whatsoever between these two variables at all. So what exactly does a coefficient of determination tell you? Well, unsurprising, you're taking that value and you're squaring it. That's it. That's what a coefficient of determination is. Now you might be like, who cares? What is the point of an R squared when we already have R? So an R squared basically will be used for linear correlations. So it would be inappropriate to do an R squared on, let's say, a exponential relationship or a logarithmic relationship or a polynomial relationship. You would only be doing an R squared with a linear relationship, either positive or negative. Now, obviously, if I'm squaring the R value, I can't get a negative R squared. R squared will always be positive. It's also going to range between 0 and 1. It effectively tells you what percentage of the variation in one variable can be explained by the other variable. That's what an R squared is telling you. An R squared value is actually, in my opinion, easier to interpret. So let's just use an example here. Let's say I have an R value of 0.5 and an R value of 0.7. It's probably not obvious that this is twice as good as this, right? Because when you square them, what do you get? It's easier to interpret the scale of differences here. This might be like, oh, it's kind of better. No, it's way better, actually. It's a big difference. So R squared is kind of almost like a easier intuitive, at least for me, able to interpret. So that's one reason that I think it's actually quite nice. And it also allows you to talk about a relationship in terms of percentages. So I think for that reason also, it's a little easy to understand. So really what an R squared value is telling you is how much better is this line versus the mean. If it's no better than the mean, that's pretty much no good relationship, right? The learning outcome here is actually exploring how can we use the coefficient of determination to actually examine what is the correlation between the size of axon diameters and propagation speed, and what is the correlation between myelination and not myelination. If we click on conduction velocity, I'm going to select non-myelinated. So here in a graph, if you see on the x-axis is the axon diameter, and that's in micrometers. So we're going real small here, right? So here is the axon diameter, and then here I have the velocity. So that would be in meters per second. So I'm going to plot non-myelinated. So there's my non-myelinated, and now I'm going to select myelinated. You could actually export this. Describe the relationship between axon diameter and conduction velocity, both for myelinated and unmyelinated axons. Both of these have a positive correlation. In both myelinated and unmyelinated, an increase in axon diameter causes an increase in conduction velocity. How are they different then? So the slope is different. So if I compare, let's say, this section and the same section, notice that for the same increase in diameter, I saw a much greater increase in conduction velocity in myelinated than I did in unmyelinated. What is the percentage difference from a diameter of 1 to 2 for non-myelinated, and what is the percentage difference between 1 and 2 for myelinated? If I want to find the percent difference here, that's 2.25 minus 3.1 divided by the average of 2.25 and 3.1. 31.8% difference between 1 and 2. The difference will be bracket 5.35 minus 10.7 close bracket divided by bracket 5.35 plus 10.7 close bracket divided by 2 close bracket gives me that times 100 gives me now a difference of 66.7%. So there's a clear, much greater percent difference for the same increase in diameter for myelinated versus non-myelinated. We see something similar being shown here. Now, if we explore these two R-squared values, notice that this trend line had an R-squared of 0.86 versus this had an R-squared of 0.77. Well, what do these values really mean? What an R-squared does is it measures the percentage that the variation in Y is being able to explain by the variation in X. Or another way that you could say this is that for a R-squared of 0.86, 86% of the variation in speed is accounted for by variation in diameter, versus for unmyelinated, 77% of the variation in speed is accounted for by variation in axon diameter. So in this way, we would say that the relationship is a stronger one for myelinated than unmyelinated given this data. Now, there's another type of relationship that we want to explore here, and that was, what is the correlation between nerve impulse and the body size of an organism? Well, in this case, we actually get a negative correlation, where the larger the body size becomes, the lower the conduction speed. Now, why might that be? As organisms get larger, they tend to have more and more complex neural pathways, and this causes a more of a delay and a lower conduction speed because there's going to be significantly more synapses or junctions between neurons. In this case, this shows an R-squared value of 0.55, meaning around 55% of the variation in conduction speed is accounted for differences in body size. Now again, these R-squared values could be based off one particular study. In order to really further examine these values, more repeats should be done in order to see, is this relationship actually this weak or is it actually stronger? And recall that just because two values are correlated does not necessarily mean they're causational. Now, we do know that these are actually causational, but it's because further work was being done to actually explain why differences in body size or myelination would impact differences in conduction speed. We've explored how neurons are propagating an action potential, right? How we went from an action potential from dendrite towards the axon and then ultimately axon terminal. What we have not discussed is how do we then move that signal from one cell to another cell. So it may surprise you to know that neurons are not directly connected, right? And that may seem odd, especially when if you're trying to compare the flow of electricity in, like, let's say, a circuit. If I don't connect two wires in a circuit, that circuit's kind of cooked, right? It means that the electrons can't flow from one place to the next. But what we do in biology and what we do in neurons and in the nervous system is that, yes, there is an electrical impulse passing through a neuron, but then that electrical impulse gets converted to a chemical signal that then passes it to the next cell to then start a brand new action potential. So that connection point is called a synapse. So a synapse is a junction, meaning a connection point, between a neuron and another cell. Now again, I don't want to make it seem that these two cells are touching. They're not. There's actually a physical space between these two cells. It's a very small space, but it's still a space nonetheless. Now, there are three different types of synapses that could occur within the nervous system. So the first synapse is actually, well, sensation. Remember, one of the requirements for being alive was the detection of stimuli, was to be able to actually interpret stimuli around you. And that could be touch, that could be pressure, that could be temperature, right? That could be a lot of things. Like, consider your senses right now, right? If touch is really just a detection in change in pressure, right? Think about temperature. You have thermoreceptors. We have pressure receptors. How does your hearing work? Your hearing actually is a pressure wave. That's what sound is. And that's being detected by cells. Your eyes, how does that work? Well, those are photoreceptors within your retina detecting changes in light. A chemoreceptor, like, let's say, smell. What is smell? It's a detection of chemicals in the air. All of these cells are what we would call sensory cells. So these are cells that are detecting some change in the environment. So this sensory cell, that's going to get activated by some change in stimulus, and then that synapses and passes that information to a neuron. So when all of your sensory cells, they're then going to pass that information to a neuron. The example that I use here is actually within the ear. If you zoom into your ear, remember, sound really is just pressure waves. And then as those molecules are vibrating, that vibration is being picked up within the very small bones within your ears, right? Those then vibrate. That vibration then gets sent to your cochlea ultimately. And within your cochlea is actually a fluid, and you have a bunch of these hair cells. And those hair cells actually will slightly move in response to sound. That movement begins an action potential. That gets transmitted, and then they will pass that signal through this synapse to a neuron. And that neuron ultimately will be joined into the auditory nerve, and that gets sent to the brain, right? So that's one type of synapse. Another type of synapse is from one neuron to another neuron. These types of connections are far the most abundant in the brain. Your brain has literally trillions of synapses, meaning connection points. And something that we'll explore in HL and in another topic as well during integration of body systems is that connection point, it's kind of weird to think about, kind of amazing to think about, but that connection point, those synapses in your brain are really what make you. That determines things like your behavior, your personality, your memories. That's all your really brain is. It's a connection point between these. And what's also really cool, that we'll discuss a little bit in the future, is that these connection points are not static. Your brain is actually constantly altering these connection points. They can strengthen them, or they can actually remove them, called neural pruning. And this is how your memory works. This is how your short-term and long-term memory works, and how you consolidate information, how you retrieve information. All of that is just synapses of neuron to neuron within the brain. And then lastly, a really important connection point, and one that we've explored in HL when we did the muscles subtopic, is if I want to send a signal to an effector cell. And an effector cell is any cell that is doing something that I want. So in our case, that's almost always going to be one of two different types of cells: muscle cells or glands. So a muscle cell obviously is going to allow movement to occur. So if I want, for instance, my skeletal muscle, my bicep, to contract right now, I can send a signal from my brain to a neuron, to a motor neuron. That motor neuron is going to synapse onto my skeletal muscle and then cause a skeletal muscle to actually contract, right? So that would be a synapse between a neuron and a muscle, something called a neuromuscular junction, something that we've explored in HL. Another one could be any organ, any structure that is mainly doing exocytosis or releasing things. Those are glands. And they're receiving signals to do this from neurons. So those are also synapses. If this is the connection point, now we're going to zoom in and actually figure out what's happening here. What's happening at this junction? What's happening at that synapse? Now the example that we're going to study in SL is something called a excitatory synapse. The root word there is excite. So in this case, this type of synapse is one where the signal is going to promote another cell to also have an action potential. Can anyone deduce what the opposite of this would be? The opposite would be a synapse where this cell is stopping this cell from having an action potential. That's called an inhibitory synapse. That's one that we're going to look at in HL. But in SL, we're only going to be focusing on an excitatory synapse. And specifically, we're going to be looking at one particular type of excitatory synapse in SL, and that's called a cholinergic synapse. Now, this term comes from the name of the major neurotransmitter that we're using. That neurotransmitter is acetylcholine. So acetylcholine is going to be the main excitatory neurotransmitter. And so any synapse that involves acetylcholine, we can call a cholinergic synapse. These are very common in these types of synapses, neuromuscular junctions. So usually, if I have a motor neuron that's sending a excitatory action potential to this cell, it's almost always going to be acetylcholine. So acetylcholine is an example of an excitatory neurotransmitter. So let's finally talk about how does this process actually work step by step. So what you're seeing here is a zoom in here. So right here, this is the axon terminal. Right here are the dendrites. So in a synapse, we can classify, we can label these cells. We can call this cell a pre-synaptic cell, as in before the synapse, and we can call this the postsynaptic cell, or after the synapse. So a synapse, in this case, is we're going to see what happens from pre to postsynaptic cell. So let's zoom in. Let's start off, first of all, as to what's happening in the presynaptic cell. So in the presynaptic cell, the first thing that's happening is, as we already talked about, an action potential is currently making its way all the way down the axon, right? So we have action, that's what I'm showing here in purple. So we have an action potential, action potential, action potential. Remember, an action potential is just the flipping of the membrane potential. So this is currently now positive, and that's from an influx of sodium ions, remember. So this influx of sodium ions are now going to cause another type of voltage-gated channel to open. In this case, these are calcium voltage-gated channels. A depolarization or an action potential caused these channels to open. So they were previously closed. If these channels open and calcium is diffusing into the cell, what are the relative concentrations of calcium inside and outside the cell at rest? They must be higher outside, lower inside, because once these channels opened, calcium diffused in. That implies that there must be some other mechanism to pump calcium out. causing that gradient to occur. So that means at rest, your neurons have a naturally low concentration of calcium intracellularly, and at rest have a high concentration of calcium extracellularly. So action potential comes down, causes the voltage-gated calcium channels to open. Calcium now diffuses into the cell. Now, calcium is actually going to act as a chemical messenger. In this case, it's going to signal this vesicle to move all the way down and fuse with this membrane. Within here, what I've shown in red, that's acetylcholine. So acetylcholine as a neurotransmitter is stored within these vesicles. Acetylcholine is now going to be expelled via exocytosis. Now, because acetylcholine, obviously, right where it's being released, the concentration is higher here, it's going to diffuse across this gap. That name of that gap, the name we can use is something called the synaptic cleft. That's just the name of the actual space between the pre- and postsynaptic membranes. So acetylcholine is now diffusing across the synaptic cleft. Okay, now it's going to bind to a different receptor now. So acetylcholine, remember, it's a chemical, and it's a chemical messenger. That's all a neurotransmitter is. So that neurotransmitter has to bind to something, and when something is specifically bound to something else, that thing that's being bound to is called a receptor. In this case, this receptor is specific to this kind of neurotransmitter. So this would be an acetylcholine receptor. So this acetylcholine receptor is going to bind this. Before the acetylcholine was bound, this thing was closed. As two acetylcholines bind to it, it's almost like a lock and key, this channel now opens. So this kind of channel, we said, was a voltage-gated channel. Same with sodium, same with potassium. That means that when there's a change in membrane voltage, that caused the channel to either open or close, right? This case, there was no change in voltage here, but there was a chemical bound to it. We can call that a ligand. So we can actually say that this is a ligand-gated channel, because a specific chemical, once bound to it, caused the channel to now be open. So this channel now opened. Well, what's always outside of the membrane? Sodium. And what happens to cells once there's a rush of sodium within? Depolarization. So that means that a brand new action potential has now been generated. Now, there is a slight problem that you may not have considered, and that is, as long as sodium keeps coming in, this signal is going to continue to go over and over and over again. And if this synapse is between a neuron and a muscle, then that's going to cause the muscle to contract forever. So if I want to stop this from happening, how do I stop this process from occurring? As long as acetylcholine is bound here, sodium is going to continue to flood in. So if I don't fix anything, I'm paralyzed. I literally can't move. So we don't want that, obviously. So your body has a natural way of stopping this process from occurring, and that is using enzymes. So I've represented this enzyme in brown here, and this enzyme, again, it looks annoyingly long, but the name actually tells you exactly what it does. Remember that the naming convention for enzymes that were catabolic, as in breaking something down, was we named them after the substrate that they work upon, right? Like amylase worked on amylose and amylopectin, right? Lipase worked on lipids. This is acetylcholine esterase. It is breaking down acetylcholine, and it's breaking it down into choline, and that gets recycled back into the presynaptic cell to be used again. So these neurotransmitters aren't being broken down and they're just deleted, right? We want to recycle them. So this then gets recycled back and then stored in a vesicle again. So as a full recap of this entire process, action potential travels down the axon, reaches the axon terminal. The depolarization wave, or an action potential, that change in membrane potential causes the voltage-gated calcium channels to open. This allows calcium to diffuse within the presynaptic cell via facilitated diffusion. This calcium influx is a term that you can use, signals the vesicles holding acetylcholine to fuse with the presynaptic membrane and expel acetylcholine into the synaptic cleft via exocytosis. Acetylcholine diffuses across the synaptic cleft and binds onto acetylcholine receptors on the postsynaptic cell. Once 2-acetylcholine have bound to that receptor, that channel opens and allows an influx or a diffusion of sodium down its gradient from outside the cell into the postsynaptic cell. That is now going to induce a new action potential to be generated, and we've now moved the signal from this cell to this cell. It's not like if I took two wires and I can actually jump a spark from one wire to the other wire. That's not what's happening, right? What's happening is I actually had an electrical impulse, chemical signal, electrical impulse again. So it's actually both electrical and chemical. I've converted that electrical signal into a chemical signal to then generate an electrical signal all over again. I think finally you're in a good position to understand why I said earlier that I cannot generate a signal in the reverse direction. Because even if I could generate an action potential here, on the postsynaptic cell, I don't have a bunch of acetylcholine being stored here. I have receptors. And on the presynaptic cell, I don't have receptors. So I can't generate the signal backwards. I can only allow it in one direction. So for this reason, this is one big advantage of having a synapse rather than directly connecting them. The other big advantage, as hopefully you guys are seeing, is that it allows control to happen. If I want to stop this from happening, I can stop releasing neurotransmitters, I can break them down, and I can stop the signal. Imagine if this cell was directly bound to a muscle, it'd be really hard to stop the signal from happening, or it would take much longer. So ironically, by having a gap here, it allows far more control and specificity. And because this gap is all being regulated by chemicals, this is really how drugs work. This is how different chemicals work. Because different chemicals can act like neurotransmitters, or they may even block these receptors. And this is kind of a whole area of biology which is really cool, which is basically neuropharmacology. It's the idea of how are chemicals impacting the nervous system. And that's a whole field of biology. Let's go over the SL learning outcomes for subtopic C2.2 Neural Signalling. The very first learning outcome that we looked at is we explored what really is a neuron and what is their cell composition. So how do they actually carry electrical impulses and the different aspects of this type of specialized cell. So students should be familiar with dendrites, axons, etc. Then we looked into how is the resting potential actually generated and maintained within neurons. How do they maintain this minus 70 millivolt resting potential? We then explored what exactly does an action potential mean? What is a nerve impulse and how is it propagated throughout an axon? And what exactly does that look like? What exactly does that mean? And that looked into depolarization, repolarization, and using sodium and potassium. We then explored really how does the speed of a nerve impulse propagation vary and what different aspects relate to this, such as the diameter size of axons and even whether axons are myelinated or non-myelinated. And this was used as a vehicle to explore R and R squared as statistical measures. We then looked at what is the junction point between two cells in the nervous system, or synapses, and how exactly they communicate with each other. And this directly relates to this learning outcome when we discussed neurotransmitters and their role in synaptic transmission. And then finally, looking at all of these together, with this one in particular, What exactly is the process of an excitatory postsynaptic potential, or an EPSP, or how can an axon transmit an electrical signal, convert it to a chemical signal to then generate a new action potential or an electrical signal in a postsynaptic cell. If you wish to learn further, which I always encourage you do, this page is a good place to start. These textbooks are those that I use and the relevant page numbers in generating these notes. So if you want to read further, I would start there. These are really great simulators at actually exploring how neurotransmission works and the function of the various channels and ions in generating action potentials. It's a really good vehicle to explore these and to simulate it using different factors. In a similar vein, these 3D models are an excellent way to actually explore these proteins and all of these various channels and pumps that are used within this neural transmission. It's really, really fascinating looking at these. And I always encourage you that you read these articles to try to get a little better understanding as to how these work. Finally, these articles are those that are a really interesting read and directly related to learning outcomes. Now, admittedly, most of these are related to the HL learning outcomes, but this one explores neonicotinoids, or a type of pesticide that is targeting the nervous system of insects. This one explores what consciousness means and why really that is really just the interaction between nervous cells. And then finally, this one actually looks into myelin and looking at it from a biological point of view and its function and its advantageous properties. Finally, I would always encourage students not just to learn for the sake of a test, but learn for the sake of learning, learn for enjoyment.